The overall goal of this procedure is to generate a three-dimensional cast of a vascular or ductal tissue. This is accomplished by first preparing and cannulating the target structure, such as the portal vein, and tying the cannula in place. Next, resin is prepared, injected into the portal vein, and allowed to cure. Then, the liver is removed and the tissue is fixed or macerated. Finally, the liver is cleared and the cast structure is visualized. Ultimately, results can be obtained that show changes in the architecture of the vascular or ductal structure through visual analysis or quantification by micro-CT.
Though this method can provide insight into liver architecture, it can also be applied to the vascular or ductal systems of any organ, including the brain, lung, pancreas, and kidneys.
To prepare the cannula, with your fingertips, warm a 1-inch long section of PE10 tubing and gently pull it so that it becomes thin. Cut the stretched tubing at a diagonal to generate a beveled tip. Cut the other edge of the tubing to create a 5 to 6 inch segment. Insert a 32 gauge half-inch hypodermic needle into the non-beveled edge of the tubing. Prepare a 3 milliliter syringe by first filling it with PBS and screw on the needle with the tubing. Ensure that there are no air bubbles in the syringe and that the needle is filled with PBS by flicking the syringe and pushing the plunger. Weigh out 0.1 grams of catalyst into a small container.